MCQs. DNA fingerprinting. DNA fingerprinting was first developed by A. James Watson, B. Alec Jeffreys, C. Kary Mullis, D. Frederick Sanger. Answer: B. Alec Jeffreys. Sir Alec Jeffreys developed the DNA fingerprinting technique in 1984 at the University of Leicester, UK. Classical DNA fingerprinting is primarily based on A. PCR amplification, B. Restriction fragment length polymorphism, RFLP, C. SNP analysis, D. DNA sequencing. Answer: B. RFLP. The old method used RFLP to detect variations in fragment lengths of VNTR regions after restriction enzyme digestion. The main regions of DNA used in classical fingerprinting are A. Exons, B. Introns, C. VNTRs, D. STRs. Answer: C. VNTRs. Variable number tandem repeats, VNTRs, vary in length among individuals and form the basis of early fingerprinting. In RFLP-based DNA fingerprinting, DNA is cut using A. DNA polymerase, B. Reverse transcriptase, C. Restriction endonuclease, D. Ligase. Answer: C. Restriction endonuclease. These enzymes cut DNA at specific recognition sites, generating fragments of varying lengths. The technique used to transfer DNA from gel to membrane is called A. Northern blotting, B. Western blotting, C. Southern blotting, D. Dot blotting. Answer: C. Southern blotting. Southern blotting, invented by Edwin Southern, is used to transfer DNA fragments for hybridization. The radioactive probes used in classical DNA fingerprinting bind to A. mRNA, B. Complementary VNTR sequences, C. Proteins, D. Restriction enzymes. Answer: B. Complementary VNTR sequences labeled DNA probes hybridize to specific VNTR sites on the membrane. The final visualization of RFLP-based DNA fingerprinting is obtained by A. Fluorescence spectroscopy, B. Autoradiography, C. Spectrophotometry, D. ELISA. Answer: B. Autoradiography. X-ray film detects radioactive signals producing the characteristic banding pattern. Which of the following is not required in classical DNA fingerprinting? A. Radioactive probes, B. PCR machine, C. Restriction enzymes, D. agarose gel. Answer: B. PCR machine. PCR is a modern addition. Classical methods did not amplify DNA. Modern DNA fingerprinting uses which polymorphic region? A. VNTR, B. STR, C. SNP, D. cDNA. Answer: B. STR. Modern fingerprinting relies on short tandem repeats, STRs, of 2 to 6 base pairs. The modern method of DNA profiling is mainly A. RFLP-based, B. PCR-based, C. Protein-based, D. Enzyme-based. Answer: B. PCR-based. PCR amplifies STR regions, enabling detection from minute DNA quantities. Which enzyme is crucial for amplification in modern DNA fingerprinting? A. Polymerase, B. Restriction enzyme, C. Reverse transcriptase, D. DNA ligase. Answer: A. Polymerase. Polymerase drives PCR amplification of STR loci. The electrophoretic separation in modern DNA fingerprinting is performed by A. Slab gel, B. Capillary electrophoresis, C. Paper chromatography, D. HPLC. Answer: B. Capillary electrophoresis. Automated capillary systems separate PCR amplified STR fragments efficiently. The fluorescent dyes used in modern fingerprinting are attached to A. Probes, B. Primers, C. Restriction enzymes, D. Template DNA. Answer: B. Primers. Labeled primers allow fluorescent detection of STR fragments. The output graph generated after STR analysis is called A. Chromatogram, B. Electropherogram, C. Spectrogram, D. Autoradiogram. Answer: B. Electropherogram. Peaks in electropherogram indicate allele sizes at STR loci. The FBI's DNA database is called A. CODIS, B. BLAST, C. GenBank, D. STR-Dex. Answer: A. CODIS. CODIS, Combined DNA Index System, stores STR profiles for forensic use. Which of the following is a major advantage of modern fingerprinting over old? A. Needs more DNA, B. Takes more time, C. Works with degraded DNA, D. Uses radioactive probes. Answer, C. Works with degraded DNA. PCR can amplify even tiny or partially degraded DNA. Classical fingerprinting required approximately A. Nanograms of DNA. B. Micrograms of DNA. C. Picograms of DNA. D. None. Answer, B. Micrograms of DNA. RFLP required large quantities of intact DNA. VNTRs are generally found in A. Coding regions. B. Non-coding regions. C. Mitochondrial DNA. D. rRNA genes. Answer, B. Non-coding regions. These tandem repeats occur in non-coding parts, hence variable but harmless. Which of the following is not a step in RFLP? A. Restriction digestion. B. Southern blotting. C. PCR amplification. D. Autoradiography. Answer, C. PCR amplification. PCR was not used in the classical RFLP method. Which labeling method replaced radioactive probes in modern fingerprinting? A. Biotin labeling. B. Fluorescent labeling. C. Enzyme-linked labeling. D. Silver staining. Answer, B. Fluorescent labeling, safer and faster detection using fluorescence. The core STR loci used in CODIS are A. 4. B. 7. C. 13 to 20. D. 3. Answer, C. 13 to 20. CODIS uses a standardized set of 20 STR loci for identification. A person's DNA fingerprint is unique because A. Genes differ. B. STR/VNTR patterns differ. C. Proteins differ. D. Mitochondria differ. Answer, B. STR/VNTR patterns differ. These regions are highly polymorphic among individuals. In RFLP, the polymorphism is due to A. Base substitutions. B. Variable repeat numbers. C. Codon changes. D. Transposon insertions. Answer, B. Variable repeat numbers. VNTR differences cause fragment length variation. Which type of DNA sample is best for RFLP analysis? A. Old bone. B. Dried blood. C. Fresh tissue. D. Hair shaft, without root. Answer, C. Fresh tissue requires high molecular weight, undegraded DNA. The fluorescent detector in modern fingerprinting is excited by A. UV laser. B. Infrared light. C. Visible light. D. Heat. Answer, A. UV laser. UV laser excites fluorescent dyes for detection. In RFLP, different individuals show A. Identical bands. B. Variable bands based on VNTRs. C. No bands. D. Random patterns. Answer, B. Variable bands based on VNTRs. VNTR differences produce unique banding patterns. The person who gave the term DNA fingerprinting was A. Mullis. B. Jeffreys. C. Watson. D. Crick. Answer, B. Jeffreys. He coined and developed the technique. The classical technique uses which blotting method? A. Western. B. Southern. C. Northern. D. Eastern. Answer, B. Southern. Southern blotting transfers DNA for hybridization. STRs are usually repeated how many times? A. 210. B. 10-100. C. 1-3. D. 100-1000. Answer, A. 210. STRs are shorter repeats, 2 to 6 BP units, repeated few times. VNTR repeats are typically A. 2 to 6 BP long. B. 10-100 BP long. C. 1 BP long. D. Greater than 1000 BP. Answer, B. 10 to 100 BP long. VNTRs are longer and more variable than STRs. The method unsuitable for degraded forensic samples is A. RFLP. B. PCR-STR. C. qPCR. D. mtDNA sequencing. Answer, A. RFLP. It requires intact DNA fragments. The unit measuring allelic size in STR analysis is A. Daltons. B. Base pairs. C. Kilobases. D. Codons. Answer, B. Base pairs. STR alleles differ in total base pair length. Which type of fingerprinting provides digital results? A. RFLP. B. STR-PCR. C. Hybridization blot. D. Dot blot. Answer, B. STR-PCR. Electrophoregrams are computer generated. Which of these samples may fail in RFLP testing? A. Blood stain. B. Old bone. C. Buccal swab. D. Fresh blood. Answer, B. Old bone. Degraded DNA from old bones is unsuitable for RFLP. Autoradiography produces A. Fluorescent peaks. B. Digital graphs. C. Dark bands on X-ray film. D. Colorimetric dots. Answer, C. Dark bands on X-ray film. It detects radioactive emissions from labeled probes. Which step was replaced by fluorescence detection in modern methods? A. Electrophoresis. B. Autoradiography. C. Restriction digestion. D. Southern blotting. Answer, B. Autoradiography. Safer fluorescent detection replaced radioactive autoradiography. The first forensic case solved by DNA fingerprinting was in A. 1984. B. 1986. C. 1990. D. 1978. Answer, B. 1986, used in the Narborough murder case, England. Which is not an advantage of PCR-based fingerprinting? A. Speed. B. Low DNA requirement. C. Works on degraded DNA. D. Radioactive risk. Answer, D. Radioactive risk. Modern methods are non-radioactive, hence safer. The final unique pattern obtained in classical method is called A. Autoradiograph. B. Kilogram. C. Karyogram. D. Spectrogram. Answer, A. Autoradiograph. X-ray image showing DNA bands after hybridization. Which statement is true about both RFLP and STR-based methods? A. Both detect tandem repeats. B. Both use PCR. C. Both use fluorescent dyes. D. Both require degraded DNA. Answer, A. Both detect tandem repeats. Both analyze variation in repetitive DNA, VNTRs or STRs. Which component is not required in old RFLP-based fingerprinting? A. Restriction enzymes. B. DNA probes. C. PCR primers. D. Gel electrophoresis. Answer, C. PCR primers. PCR was not part of old RFLP, only restriction digestion was used. What kind of DNA markers are STRs and VNTRs? A. Codominant markers. B. Dominant markers. C. Recessive markers. D. Epigenetic markers. Answer, A. Codominant markers. Both alleles from parents are visible in results codominant inheritance. Which type of DNA fingerprinting allows degraded sample analysis? A. RFLP. B. STR-PCR. C. SNP-seq. D. Restriction mapping. Answer, B. STR-PCR. STRs are short and amplified by PCR, suitable for degraded forensic samples. The number of STR loci used in CODIS for human identification is A10, B13, C20, D23. Answer, C20. CODIS now uses 20 standard street loci for high accuracy. Which Indian city hosts the National DNA Database Center? A. Delhi. B. Hyderabad. C. Mumbai. D. Bengaluru. Answer, B. Hyderabad. The CDFD in Hyderabad functions as the National DNA Database Hub. Which statement is true for RFLP-based fingerprinting? A. Requires PCR. B. Uses fluorescent markers. C. Detects length variation after restriction enzyme digestion. D. Uses mitochondrial DNA. Answer, C. Detects length variation after restriction enzyme digestion. RFLP detects fragment length polymorphisms at VNTR regions. Which year did India officially begin forensic DNA fingerprinting services? A. 1984. B. 1988. C. 1995. D. 2001. Answer, B. 1988. India began forensic DNA testing at CCMB under Dr. Lalji Singh in 1988. Which type of sample is least suitable for RFLP analysis? A. Fresh blood. B. Hair root. C. Old bone fragment. D. Tissue biopsy. Answer, C. Old bone fragment. Degraded DNA in bones is unsuitable for RFLP, but usable in PCR-based STR. The statistical probability of two individuals having the same DNA profile is A. 1 in 100. B. 1 in 1000. C. 1 in 10 million to 1 in 1 billion. D. 1 in 10. Answer, C. 1 in 10 million to 1 in 1 billion. The high polymorphism of STRs makes matching profiles extremely rare. In forensic science, DNA fingerprinting helps to A. Diagnose diseases. B. Identify criminals and relationships. C. Cure genetic disorders. D. Modify genomes. Answer, B. Identify criminals and relationships. DNA fingerprinting is vital for criminal identification and paternity verification. Thanks for watching.